Hello, ladies and gentlemen. My name is Jason David Klesak. I am from Phoenix, Arizona, and I'm 33 years of age. I am an avid enthusiast in the sciences of many fields of science, organic chemistry, business, molecular biology, cellular biology, microcellular biology, medicine, nuclear medicine, just to name a few. The list goes on and on. I have been an avid enthusiast of these sciences for many years, since my youngest of ages. Just one moment, I need to pull up a menu. So I postulated that these 5 receptor sites, they have an activation key of some sort, and utilizing this software Nanome that I have been somewhat tinkering with, but not too much, not too often. It's a tool provided in virtual reality. As you see, Nanome right there. So with this postulation, I theorized that these 5 keys in some way would be visible utilizing cross-analysis of pharmaceuticals, such as Fenoldopam, Dopamine, levodopa, zolpidem. I compiled a list from drugbank.com as well drugbank.org of drugs that target specifically one or more of these receptor sites. As you can see, here is one of the sheets that I compiled with the data. Here in this column right here is drug bank ID, the name of the pharmaceutical drug group, if it's been approved, is it investigational, is it vet approved, is it withdrawn. In this column right here is pharmacological action. Does it have confirmed pharmacological action or not on this receptor site? Yes or no, or unknown. In the actions column lists what type of action occurs: agonist, antagonist, partial agonist, blocker, binder, inhibitor of some sorts. For this solution, or for the answer to this, what some would say is a problem, a question is, what do these look like? I compiled, and over the course of about 100 hours of manipulating, folding around, I was able to finally get a picture of, with near exact, almost perfectly aligned, an atomic arrangement of what these keys look like. I got D1 alpha right here. This is the D1 alpha key. This will activate agonist activity at D1 alpha. This is D1 Bravo key. This will activate agonist activity at D1 Bravo D5, and so on and so forth, D2, D3, D4. So in this presentation, I'm going to go over a few things, such as confirmation, as well as showing where these keys fit on the medication and why and how I know for sure that it's these and not any other arrangement. So first I'm going to start off with Fenoldopam. And we're going to go on to Amantadine, Rotigotine, Pramipexole, and take a look at Apomorphine as well. So starting off with Fenoldopam. Fenoldopam is from a D1 and a D5, so D1 alpha and D5 Bravo activator, agonistic activators. Now, based off of my cross-examination and analysis of Fenoldopam, it has been reported with confirmation that it has agonist activity, pharmacological action, yes, at D1 and D5, with no activity reported or is currently unknown. So without further ado, this is Fenoldopam right here. You've got 2 oxygens, a single oxygen, a nitrogen, some carbons. And here is the first overlay. So D1, Bravo, and D5. Fluoride. So there is the D1 overlay. Here is the D5. Now, placement of the arrangement of the hydrogens is rather quite important. As you can see with Fenoldopam and the sheet, we've got reported pharmacological action, yes, actions agonist at D1 and D5, D1 and D5. And just for comparison of the keys, the receptor activators, I'm going to attempt to overlay these other 3 keys onto this molecule. No D3 key on Fenoldopam. No D2 key on Fenoldopam. No D4 key on Fenoldopam. The only 2 keys that overlay with near perfection, and this is as close as I could get them pretty much. Utilizing my own mind. Next on the docket is Amantadine. Here we have Amantadine, a single spindle crystal. It seems almost looks like a dreidel, somewhat. As you can see, it's a very simple molecule. Now, once again, for your visual comparison, I'm going to attempt to overlay and show you each of these. I can line up a few of these carbons with this key, and the bond doesn't seem to be too the overlay lineup, the atomic arrangement of the key for the receptor slot based off of my analysis has to be near exact in position relative to the receptor. So we're talking very narrow margins of error when formulating these keys. If it's off even by this much, then it won't fit into the lock, so to speak. And you will get either A, low binding affinity if it's near it enough, but if it's too far outside of the margins, you'll get no binding affinity. Here is the D2 key, and Amantadine has only agonistic activity, pharmacological action, yes, at receptor site dopamine receptor site 2. So as you can see, it has a near perfect overlay of the amantadine molecule. Now, if certain metabolisms line up, there is potential for the D4 key to activate agonistic activities while in the receptor site. There has been currently no investigations into amantadine for D4 agonistic activity. Has not been investigated or tested as of yet. So as you can see, there is a single key and I can confirm with surety that it has potential for medium to high binding affinity at the D4 receptor site as well. Next on the list we have Rotigotine. Now the angle of the planes, we'll just call this plane molecule plane 1, molecule plane 2, molecule plane 3 right here. Rotigotine. Rotigotine is approved with pharmacological action, yes, with agonist activity at receptor sites 2, 3, and 4 confirmed, with D1 Bravo being unknown, but currently pharmacological action unknown, but currently being investigated or approved without investigation. So here is, once again, for your visual comparative analysis as well, D3, D4, based off of the comparative analysis that I conducted, the nitrogens here, the atom in this location, in these locations, is not as important for receptor site activation as is the spacing of the atoms in the atomic arrangement of a particular atom, the spacing from from the carbon and nitrogen. See how the distance between here and here, for instance, if I try to overlay this way, it's too deviated from the margin of error necessary for receptor site activation. But if I rotate it like so, as you can see, the overlay is near perfect. Now I'm going to overlay the D4 key. And I'm also seeing a D1 Bravo key right here. Now, D1 Bravo has not been confirmed with a pharmacological action of yes, with agonistic activity, Rotigotine. But it is currently approved with pharmacological action unknown agonist for receptor site D5. Now, I do have to state that I did not utilize, as I wrote this up here as a somewhat of a disclaimer, with the color-coded chart, rated dopamine receptors 1 to 5 keys, activators of agonists activate receptor sites, only atomic arrangements with pharmacological action of yes and agonist only were utilized during the solvation process. No agonists with pharmacological action of unknown were utilized during the solvation process, which is so I can confirm that rotigotine does in fact and will have pharmacological action of an agonist with near perfect binding affinity at receptor site D1 Bravo. Now, the further these deviate in the medications, so by the size margin of errors are pretty narrow. We're talking 0.1%, 0.2%, in terms of arrangement alignment of the keys, or arrangement in the receptor sites. Next on our list we have Pramipexole. Pramipexole. Pramipexole is approved investigational pharmacological action, yes, with agonist activity at dopamine receptor sites 2, 3, and 4. So once again, for your visual comparison, as well, your visual analysis, I will attempt once again to overlay these 5 keys onto Pramipexole. Now, as the amount of error increases from the margin of error of the arrangement of the molecules, or of the keys in the molecules, the farther out you get from the margin of error, the lower the binding affinity, and when you get too far out, it stops binding it has no binding affinity at all. Bonding affinity. And last but not least, we have Apomorphine. Okay. D2, D3, D4. I am seeing no overlay for the D3 key. I'm also seeing as you see, a D2 key, which I didn't use the unknowns during the solving process. But it has been approved Apomorphine approved investigational pharmacological action unknown at this time. I can say with absolute certainty that it indeed does have pharmacological action, yes. Agonistic activity. I'm going to place these back right in their locations. And I'm going to show everyone a close-up of each. And this will conclude my presentation. I was unable to acquire with exact accuracy the amount of hydrogens necessary at the end of each, or at each atom, or how many, 1, 2, or 3. But, and it's with all of them. A large percentage of the hydrogens that I was able to figure out are indeed in their what some would say their correct positions and amounts. I was unable to ascertain the amount of hydrogens per atom for each of the keys due to the vast differentiation or vast differences in the medications in terms of hydrogens. As well as angle. But the arrangement of the atoms is close to 100% accurate. In terms of their position at nanoscales, or atomic scales. Ladies and gentlemen that have viewed this presentation, I'd like to thank you for your time and your consideration, as well as your careful thought in matters pertaining to things such as this. I hope you have yourselves a wonderful year, and this concludes my presentation of the 5 keys.